Okay, so on Friday we were talking about myoglobin and how it binds oxygen, and we were starting to make a segue into hemoglobin in contrast to myoglobin, which is simply to store oxygen. Hemoglobin has to transport oxygen. So today we're going to, in terms of thinking about structure and function, look at the structure of hemoglobin, its quaternary structure, look at how the oxygen binds to the heme, what the consequences of that are, and how that facilitates what hemoglobin has to do, which is to transport oxygen, which means to bind it initially in the lungs and then release it later. So we'll look at some of these structural changes that happen when oxygen is bound. We're going to look at a series of interactions between an FG corner of one subunit and a C helix of another. And if you've looked at the notes, you know, there are a lot of salt bridges and so forth. The important thing when we get to that, you're not going to have to memorize all those. You should basically know that as it goes from the oxygenated form to the deoxygenated form, there are changes in these. And there's one interaction which I'll highlight that is important for you to remember. And that will come into play when we talk about the Bohr effect, which is one of two examples where the binding of one ligand is going to affect the binding of oxygen. All right, so oxygen is having an effect on the affinity of hemoglobin for itself. That's the basis for cooperativity, but we'll also see how other things which can bind to hemoglobin, pH meaning protons, or BPG, how their binding affects the affinity that hemoglobin has for oxygen. And then we're going to talk a little bit about hemoglobin genetics and sickle cell anemia. Okay, before we do that, myself, Drs. Granosowski and Surtees are looking for anyone who has work study, is work study eligible, preferably already has their work study letter, to come into the lab and basically work. All right, so you come in with the work study, and it varies, I guess, between the different faculty what they'll have you do. Generally start out with general lab stuff, see how good you are, and if you can transition up to doing experiments, great. All right, so if you're interested and you are work study eligible, and if you already have the letter, which is preferred so you can start quickly, you can contact one of these people. Okay? So, central issue today, we want to understand the structural basis for the cooperativity that hemoglobin has for oxygen. We talked about the binding curves and how that sigmoidal binding curve was reflective of cooperativity, and that was exactly the type of behavior that we would like a transport protein to have. So we want to understand what the structural basis is for that cooperative binding, and then, like I said, we'll talk a little bit about globin genetics and how they affect hemoglobin function. So on Friday, what did we say about the inter-subunit contacts? I told you that you should think about hemoglobin as a dimer of heterodimers, that when you look at the contacts between these, that the contacts between an alpha and beta and an alpha and beta, that those contacts are so strong that they really can't be changed. All right? The ones which are altered are these transverse ones between alpha 1 and beta 2, beta 1 and alpha 2. And those are the ones that we're going to be looking at both pre-oxygen binding and post-oxygen binding. But you should just remember that if this molecule that has quaternary structure is a dimer of dimers, that if there's going to be some movement, what we're going to see is the movement is with respect to these dimers towards each other. Okay? All right, so the first thing that you should appreciate is that the deoxy form and the oxy form, and we also refer to the deoxy form as the T state, the oxy form as the R state. All right, I'm going to kind of give you a take home at the outset and not make it quite so much a mystery so that you know where we're going. All right? When you look at the deoxy state, this deoxy state has this large cavity. This is a top view, this large central cavity that's aqueous. You have a situation of those two dimers, those alpha-beta dimers relative to each other. When it goes into the oxy state, you see evidence for the change in the quaternary structure. That solvent-filled cleft here is smaller, and you have a rotation of these two alpha-beta heterodimers relative to each other. All right, and you can see that in the notes. The important thing is to realize that between these two, there is a global change in the quaternary structure. To kind of give you the take-home to see where we're going, is that in this state, right, the deoxy state, there's no oxygen bound. In the oxy state, the conformation of that hemoglobin, the quaternary structure of that hemoglobin, is exactly the same if you have one oxygen bound, or two, or three, or four. All right? So what you should think about hemoglobin is really being in only one of two states. One is the deoxy, no oxygen. The other is the oxy. And to kind of get you thinking ahead of where we're going is, let's say we're in the deoxy state. If we bind one oxygen molecule, if we're successful in doing that, that's going to trigger that change in the quaternary structure. And that structure is then going to make the remaining binding sites for oxygen in the preferred state for binding oxygen. Okay? And that's why we're going to see cooperative binding. We'll go back over this, but just think about deoxy in one quaternary, binding of one oxygen triggers this transition, and then when it's in this state, it's in a much better state to bind more oxygen molecules. All right? The basis of cooperativity, we will come back to look in greater detail. So this is just another way. This is looking at it from the side, and it just shows this 15-degree shift. So we've got that shift of the heterodimers relative to each other. We have a change in that solvent-filled cleft, but the most important thing is it's a global change in quaternary structure. So let's think about that just for a second. Remember, we said that when you have cooperativity, if you bind one ligand, it has to have a means to relay that information to the other ligand binding sites. So that's, in fact, what's happening here is if one oxygen binds and it triggers this global change, the hemes on those other three subunits know that an oxygen has bound and there's been a structural consequence. They themselves then get into a conformation which is more conducive to oxygen binding. And that is what makes the affinity go up. So as you bind one oxygen molecule, the affinity for binding more oxygen increases. All right. So what's happening here? Right now, before we get into that first step of the transition, it's important to appreciate that there are, in this shifting, right, if an alpha-1, beta-1, and alpha-2, beta-2, those heterodimers, if they are maintaining their integrity and we're just getting a shift, that the shift is going to cause changes in these, what we'll say, transverse interactions. So how many subunits are in hemoglobin? 4. So if we look at, let's say, these two, upper left and bottom right quadrant of the tetramer, that where they come into greatest proximity is, here's the heme, and then this is a beta subunit, and what do we say FG was? That's going to be the corner between the F and the G helices, and it is interacting with the C helix here of the alpha 1. So we have these interactions. Because of the symmetry here, we have the C helix of this beta chain interacting with the FG corner of that alpha. So that's how many sets of interactions? 2. Then we have the other 2 subunits doing the same thing. So we've got 4 sets of interactions at the close contacts between these FG corner and the C helices. If you look in the notes, or if you look here we've got this set, this set. In blue, you have those interactions that are going on in the deoxy configuration, and in red in the oxy. So if you look in your notes, you'll see that there are some interactions that are maintained, right? These are hydrogen bonds, salt bridges, some hydrophobic interactions, and some of them persist in either state, but some of them are unique to either the oxy or the deoxy. It's not important to memorize all these interactions, all right? What you need to realize is that these interactions do change as you would expect that they would change because of the change in the conformation, all right? We will come back to this histidine residue here at this C terminus. This one you should remember, because it's not only involved here in this overall stability, and if you look at it in blue, right, we've got a protonated histidine that is helping stabilize the deoxy form, and we're going to come back to that when we talk about the Bohr effect. So know that there are a lot of interactions here. Some are maintained, some are abolished and then formed in the altered, in the opposite configuration or the opposite form. But that is to be expected because of this change in quaternary structure. All right, so how does this happen? Well, this is just another way of looking, kind of an easy view of what's happening. So here we have a histidine in the FG corner. We've got an aspartic acid, okay, and that is involved in what kind of interaction with this tyrosine? It's a hydrogen bond. You have an OH group here. And all this is meant to show you is that upon oxygenation, we have a shift in the relative position of this subunit versus that subunit. So the FG corner C helix interactions, they're not completely abolished. We just get a shift. Okay, and now we have a new hydrogen bond going on between these 2 residues. Okay, so the oxy form and the deoxy form, they have their own set of unique interactions that are helping to stabilize those, right? But obviously the binding of oxygen is having an effect on which configuration you're going into, and these other 2 allosteric effectors, protons and BPG, we'll see how they work later. All right, so here's where we're going to start to think about what does the molecule or what does the ligand binding site look like prior to binding oxygen. What did we say when we were looking at myoglobin and we were talking about the structure of the heme ring prior to and post binding of oxygen? We said that when the oxygen binds, it flattens out. So here we are in the T state, the deoxy state. So our iron is here, the heme ring is kind of puckered, and so there are 4 ligands here, right? When we say 4 ligands of iron, the 4 nitrogens of the heme ring, we said that it was bound on the backside to that proximal histidine. Remember that? The oxygen is going to come in where? It's going to come in here. So when the oxygen binds here, it causes this heme ring to flatten out. And that causes this histidine, which is covalently bound, to shift into this plane. There's an 8-degree shift here. Okay? Now, when it does that, you then get strain introduced, most notably here, where there's a valine that is on this FG corner. So when oxygen binds, then when this flattens out, this strain has to be relieved. We're starting to get steric effects here. And so what happens is, as after it binds, to relieve this strain, this valine FG moves. Now, if the valine on the FG corner is moving, then that tells you more than just the corner is moving. Right? So the initial thing to realize is that oxygen binding, when it does that, it's going to induce a conformational change, because when it binds the heme flattens out. When the heme flattens out, it's connected to the thigh bone, etc. It's connected to this histidine. The histidine's going to move, but then it sees that there's a valine sitting there, and it says, I've got to move because the heme bound oxygen, the heme is flattening out, I've got to move with the heme, you're in the way, move. All right, and in that way, it's going to start to trigger a whole series of conformational shifts. So this is just another way of looking at that, just that the heme in the deoxy versus the oxy, right? In the blue is the deoxy. It binds oxygen, it flattens out. We're getting steric effects now with this valine, okay? So you basically are going to break a hydrogen bond as this valine 98 moves, all right? This valine moves, and what that does is it causes a hydrogen bond to be broken, and actually, as that is broken, then that starts to destabilize the structure, and it starts a cascade of events. Okay. So, what did we just say? The oxygen is binding to the iron in the heme. As that iron moves into the plane of the porphyrin ring, it's flattening out, and that proximal histidine on the other side wants to move with it. So with that shift, okay, that F helix of beta 2 is moving closer to the heme ring. That causes that FG corner to move. As we showed, that valine has got to get out of the way. So we're getting hydrogen bonds that are broken, new ones that are made, salt bridges that are made, salt bridges are disrupted. But because of that steric shift, that oxygen now has bound, but the rest of the molecule now is going to sense that that has happened, and why. The oxygen's binding to the heme, okay? Once it binds, we've got to get some movement. That FG corner has to move. So that beta 2 wants to move relative to the C helix and alpha 1. But what did we say about the structure in terms of it being a dimer of heterodimers, okay? It can move like this, but no one subunit can move independent of the others because they're linked by those strong interactions, right, between alpha 1, beta 1, alpha 2, beta 2. So if If we've got inflexibility in alpha 1, beta 1, alpha 2, beta 2, then if you get binding of oxygen on one heme and it's saying, I want to move my FG corner relative to a C helix to alleviate that strain, if I'm going to do it over here, that subunit can't move independently of who it's linked to. So then that has to move, and as a result, you're either going to get all or nothing, right? Either everybody's going to move or nobody's going to move. Do you understand that? All right? So if everybody moves, then this TR shift must occur simultaneously at both of these interfaces. So when you look at the heme, for oxygen to bind, right, that histidine has to move over with the heme ring flattening out, and that FG valine has got to move. If it does it on one, if it's successful on one ligand binding site, then for it to be successful, we have to get movement, but that movement there has to be accompanied by movement in the other three subunits. So that triggers them all to shift in a similar fashion. So those other three oxygen binding sites, right, what kind of structure are they in? They're in a structure where the FG helix, or the FG corner relative to the C helix, has already moved, almost as if it was anticipating there would be strain. So there's no strain there now, and if an oxygen comes, is it going to have an easier time, the same, or a more difficult time binding to that heme ring? It's going to be easier. So when you're thinking about cooperative binding, right, structure and function, we go back to the beginning. It wants to be a transport protein. To be a transport protein, it has to change its behavior. It has to be schizophrenic. It has to be a high affinity oxygen binder in the lungs, and then somehow something has to change when it gets into the tissues. It has to be able to go between two states, and those two states are T and R, and you should see how the binding of oxygen here affects that transition. Okay? All right, so as I just said, the binding of one oxygen is accompanied by a global quaternary structural change. You're not getting a change in the structure just at the immediate vicinity or locale of that first binding event. You're getting a global change in the entire molecule. All four subunits are undergoing structural changes, even though you've only had one binding event. So these unliganded hemes in the R state, they've got a higher affinity for oxygen. So they're sitting there, and basically the first binding event kind of primed the pump, caused the conformational change, and now the other three unliganded sites have a higher affinity for oxygen. So just think about hemoglobin existing in one of two forms, right? The T form or the R form, where all the binding sites for oxygen are in a productive conformation or a non-productive conformation. Is that pretty clear? All right. So, oxygen binding to hemoglobin to affect subsequent oxygen binding is what kind of an effect? It's an allosteric effect. Remember, we talked about allostery, and it is a homotropic allosteric effector. The binding of oxygen affects the binding of oxygen. So here we're going to talk about oxygen binding that's regulated by hemoglobin binding something other than oxygen. All right, so the Bohr effect is the pH effect on oxygen binding. So whether a proton is bound or not on there is going to affect it, and then BPG. All right, so let's say you look at binding of oxygen at different pHs. So here's myoglobin. What kind of curve is that for myoglobin? Rectangular hyperbola, right? That curve that we see there is classic. It's a classic binding curve for any protein binding something. We see with hemoglobin, right, because it goes from two different binding states, two different binding affinities, it's sigmoidal. So if you look at the binding of oxygen as a function of pH, all right, you get these different curves. So here, 50% saturation, theta, we call the P50. So what's happening to the P50 as you go to lower pH? As we go from pH 7.6 to 6.8, what's happening to the P50? If the P50 goes up, that means we need a higher concentration of oxygen to have 50% saturation. So if the P50 is higher, does that mean that oxygen is being bound more tightly or more weakly? More weakly, right? Higher P50. So why does that make sense? And why does it want to do that? Right. So the target for the oxygen delivery is the tissues. And in the peripheral tissues, they're pumping out CO2 into the blood, all right? And that CO2 is lowering the pH in the blood. So in contrast to the lungs, the blood around the respiring tissues are pumping out CO2 to lower that pH. So when the hemoglobin now is coming into there, it's going to release it. We can see that from the curves. What's the structural basis for it? When we talked about basic side chains and acidic side chains, in the physiological pH range, arginine and lysine are always going to be positively charged, right? We can talk about an experiment or an in vitro, purification of the peptide and hypothetically say, well, we're going to do it at this pH, this pH, this pH. But in terms of physiology, because of the pKa's, the basic side chains of lysine and arginine are always going to be positively charged. Aspartic acid, glutamic acid, negatively charged. So, I'm Italian, so I probably shouldn't say this, but the Italians were initially with the Nazis and then they moved with the Allies. So, there's one basic side chain that kind of can go either way, and that one is histidine. And it depends upon its environment. If it's surrounded by Germans, all right? We're lovers, not fighters. So that histidine, if it is in a low pH environment, is it going to be protonated or neutral? The lower the pH, remember, second lecture, acid-base behavior, it's going to affect charge is going to affect, if that histidine is in a hemoglobin that goes into the tissues and the pH there is lower, the state of that is going to be protonated. And it's going to have a positive charge, and what's that positive charge involved in? It's involved in a salt bridge that stabilizes what form? The deoxy. So if something is stabilizing the deoxy form, if it's stabilizing that, then is that causing the affinity for oxygen to go up or to go down? Down. So the Bohr effect is dealing with the pH effect on the protonated versus unprotonated state, what? Of that histidine. And that is going to affect whether or not you have that salt bridge stabilizing the deoxy form or not, and that's going to have an effect on the oxygen affinity. So as you get to lower pH in the tissues, oxygen is going to be released more readily, and that's what we want to do. All right? That ligand, in terms of a proton, that allosteric effect is kind of rapid, meaning in very short periods of time, you're going from one state to the other, right? Binding the oxygen in the tissues, or in the lungs, releasing it in the tissues, all right? The second allosteric effector is something that is there in the red blood cells all the time, and it's having a more long-term effect on oxygen affinity. Remember I talked about, you listened. The top view here, and we had this water-filled cleft that we said in the deoxy form was larger than in the oxy form. So BPG is in the red blood cells, and the concentration that it's there can vary. I'll mention why. But if we look at the structure, BPG fits really nicely into that solvent-filled cleft of the deoxy form. So it fits in there, and itself, BPG, is this polyanion. Okay, so it has these negative charges. And in adult hemoglobin, what do you have projecting into that cleft? Okay? You've got positive charge here, positive charge here, a histidine here, a histidine here. All right? So with all of those positive charges projecting into that cleft, and that cleft can fit a polyanion, we have a series of ion pairs, salt bridges, and that is stabilizing this structure, right? This structure, where the solvent cavity is large enough to take BPG in, is that the oxy form or the deoxy form? It's the deoxy form. So in the deoxy form, BPG can get in there, have stabilizing interaction. So if BPG stabilizes the deoxy form, what does that do to the oxygen affinity? It decreases. So it's going to cause oxygen to be released more readily. So what is it that BPG, why do we want to have that in the red blood cells? I'll give you one example in terms of oxygen transfer between maternal hemoglobin and fetal hemoglobin. But let's say you guys decide to drop out of school and become ski bums, and you move to Aspen, Colorado. Not a bad idea. No, I'm only kidding. The altitude there, right? The oxygen is lower. Your oxygen delivery to your tissues is going to be down, right? And actually what happens is you can still hold on to oxygen, but BPG will cause, in other words, you'll still be able to bind oxygen there. If your BPG in your red blood cells is increased, then you're going to have some negative effect on binding the oxygen in the lungs, right? But you have a much greater beneficial effect on oxygen release to your tissues. So the oxygen in the air is lower, but if your red blood cells have more BPG, you're still going to be able to bind most of that oxygen, but then you're going to be better at releasing it. So BPG, then, the amount in your red blood cells would change over a longer period of time, right? It's a kind of a more general switch for how efficiently you release the oxygen. Right, so a better example of that is probably looking at the transfer between maternal hemoglobin and fetal hemoglobin. So in terms of the genetics, adult hemoglobin has 2 alpha and 2 beta chains, right? In fetal hemoglobin, you still have the alpha chains, but these gamma chains are the ones that are expressed. So adult form is alpha 2, beta 2. Fetal form is alpha 2, gamma 2. Right? The structure and everything is very, very similar. So there's switching going on in terms of your genetic expression. During fetal development, this is the form of hemoglobin, and then as you become an adult, this is. All right? So we can ask why in a minute. The other point, and we're not going to get into the specifics in terms of the disease and the structural basis of the thalassemias. There are many diseases which are caused, right, by mutations that cause a host of structural changes that result in functional changes. All right, we will talk about one in terms of sickle cell. But for right now, in terms of thinking about BPG, BPG binds to the deoxy form and it decreases the oxygen affinity. So let's say you're a developing fetus, and where are you getting your oxygen? You're getting it from your mother, and really if the mother's hemoglobin is holding on to the oxygen, then how efficiently is that transfer going to be to the fetal hemoglobin? It's not going to be very efficient unless you give an advantage to the fetus. So because of these gamma chains, when you look at the structure of fetal hemoglobin, okay, there are 2 changes. First, this is simply showing what you would hope to see, right, that the fetal hemoglobin, right, has a lower P50 than the mother's. It's a better binder. It has a higher affinity for oxygen, and that is because the BPG that would normally be in the adult hemoglobin, it is there in that cleft and interacting electrostatically with these positively charged groups, and it's a polyanion. In fetal hemoglobin, this cleft is significantly smaller because of the structural changes in those gamma subunits relative to the beta subunits. And also what's projecting into the cleft are hydroxyl groups or neutral groups that don't have that positive charge that you had before. So all things being equal, structures being fairly similar, the inability for fetal hemoglobin to stabilize binding of BPG means the BPG is not going to be able to get in there and stabilize the deoxy form. So if it doesn't do that, then what is the resulting change in the oxygen affinity? It's going to go higher, okay? So the basis for that oxygen transfer is that you have a higher affinity for oxygen with the fetal hemoglobin. Okay, sickle cell anemia. So in the beta chain, okay, you have a mutation. The glutamic acid that is normally at position 6 has been converted to a valine. So in problem sets, we'll say, you know, which of these are going to be the least dramatic changes on function or most dramatic. If you look at this, that was dramatic. The glutamic acid is it's negatively charged, right? And you're going to valine, which is nonpolar. So that's a big change, right? You're going from something very hydrophilic and charged to something that's hydrophobic. So that side chain of glutamic acid 6, not surprisingly, is normally on the surface, exposed to the water. So now you are throwing in a valine. So is that good or not good? It's not good. All right. Now we'll look at this, but because of that amino acid substitution, two things happen. One is that you've got a hydrophobic residue at a place that it doesn't really like to be, or maybe it doesn't really care, but the water molecules don't like it there. The second is that when you look, remember we have the two conformations of hemoglobin, the oxyform and the deoxyform, and there's a structural change. In the deoxyform, and this is important, not in the oxyform, there's a little complementary hydrophobic patch that is formed, all right? And what happens as a consequence is that valine, which is normally interacting with the water and all of that's unfavorable, it's looking for a refuge somewhere. And as a consequence of this mutation, in the deoxyform, you have a little complementary patch that's formed over here. Okay? So remember we have two beta chains, right? And two alpha chains. So for hemoglobin S or hemoglobin sickle, both subunits have to be mutant, right? They're coming off the same gene. Okay? So this subunit has this valine being exposed, and this is in the deoxy configuration that you have that pocket. So in the oxyform, there's no real structural consequences of this mutation. But it's in the deoxyform, in the T state, where with two mutant subunits, you have two structural manifestations. One is this valine at the surface, and another is this little hydrophobic pocket. So what happens is we start to form these fibers. Right? So here you would have this hydrophobic pocket, and this hemoglobin molecule, and this is in the deoxy form, after it's given up its oxygen, this valine now is going to interact through hydrophobic interactions with that pocket. And then that pocket with that valine. That pocket with that valine. All right? So we're going to get these fibers, and this is like the structural model of it. But here, okay, is actually showing you a sickle cell red blood cell where you can see these fibers in it. So when we talk about sickle cell, you'll hear sickle cell anemia. So there are three consequences. You've got one amino acid chain, or one amino acid change. That amino acid change within a multi-subunit hemoglobin causes two structural changes, which it is on the surface, but at first glance you wouldn't think, well, this is not going to wreak havoc. But in the deoxy form, you start getting these polymers, and those polymers then do two things. The first thing it does is it causes the shape of the red blood cell to sickle. It causes it to go from nice and round to this sickle shape. Do these sickle cells exist when they are oxygenated in the blood, in the lungs? No. It's when they've gone to the deoxy form. And when they've gone to the deoxy form is when they've given up their oxygen. They give up their oxygen at low pH, and the low pH is where? In the tissues, and as it goes further and further into the tissues, you get into the capillaries, and that's where it's being released. So with the narrow capillaries, it goes in as a spherical hemoglobin, being a spherical red blood cell being carried by a spherical red blood cell with the hemoglobin in. The hemoglobin gives up the oxygen, the fibers start to form, the cell changes its shape, it's trying to come out of the capillaries, and what happens? They start to hook together and click together. So that's where the pain of sickle cell anemia comes from, is the lodging of these abnormally shaped cells in the capillaries. Where does the anemia come from? The anemia comes from, as these fibers form, you're starting to make these little prickly pairs inside the cell, and they start to lyse. And as you lyse, you start losing red blood cells and you become anemic. Right? So all of that is coming from removing a glutamic acid side chain and putting on a valine side chain. Right? And because structure is imparting function, and you've got a multimeric, a multi-subunit thing here, the structural changes end up actually being significant enough that it gives different properties to the molecule. In all of the stuff that we talk about, structure dictates function. You've got a normal structure, it has normal function. You start making mutations, and function and structure, well, you know that the structure has the potential to change. If it does, then the question is what's happening with the function. And in all of these diseases, you may hear about them, misfolding, this, that, Alzheimer's, but really when you get down to it, it's chemistry, all right? So, so just to summarize, all right, hemoglobin is interconverting between these two global states, R and T, through changes in their quaternary structure, and they have different affinities for oxygen. We know that that's regulated by oxygen itself, which is homotropic allosteric, and then the other physiological important ligands are protons in the Bohr effect and 2,3-BPG. And then we didn't get into the thalassemias, but there are a whole host, okay, of blood disorders which are due to mutations in the hemoglobin subunits, okay? All right, so we've got a couple minutes, hold on. So the first exam is a week from today, and the material that will be on the first exam is through tomorrow. Tomorrow's lecture 8, right? So after tomorrow, I wouldn't just start studying and not come to class on Thursday and Friday, because lectures 9 and 10 are going to be on the next exam, and those are important, all right? But just so that you know, problem set 2, I will post that early today, and problem set 2 is going to cover lectures 5, 6, today, and tomorrow. So we will go over that on Wednesday in recitation. Now, the question, how many of you realistically can make the 2-hour recitation on Wednesday? I mean, really, all of you should be able to. How many can't? Okay. Not too many. So what I would like to do is we will have recitation on Wednesday, and we're going to focus only on the first 8 lectures, because we wouldn't have had the 9th yet. Problem set 1, problem set 2. We've got the room for 2 hours, but in fact, I think it can actually go maybe till 12. So recitation on Wednesday is essentially going to be the review for the exam. All right. I will also put up some practice exam questions, but they are essentially the same as your problem set questions in terms of the severity of them. All right. See you tomorrow.
I have the work study letter. Should I just email you, or can I bring it tomorrow?